So today we're going to look at the scientific field of drugs. The first aim is, can you describe the 4 general types of drugs and their uses? Then can you evaluate claims on specific drugs? And then can you explain the key stages involved in drug testing? I think it's reasonable to assume that this topic has relevance to every living person on this planet. I think the word itself, drugs, has a strong impact on the listener. It's very hard to separate the word drug from the idea of drug abuse. However, as you'll learn, drugs have many applications, not all of them harmful. So let's start off by defining the term drug. A drug is a chemical that will interfere with chemical reactions that occur in the body. Largely chemical reactions which occur within the nervous system. Some drugs can be addictive. That means they interfere with a network of neurons in your brain which are called the reward pathway. When impulses flow through your reward pathway, they make you feel good and encourage the repetition of that behavior. Such drugs include heroin, caffeine, nicotine from cigarettes, and cocaine. Repeated use of addictive drugs can lead to withdrawal symptoms. These are very painful side effects people experience when they're deprived of that drug. Some can be incredibly dangerous. There are 4 general categories of drugs. One category are referred to as painkillers. You may have heard of paracetamol. That's a very readily available drug you can receive over the counter without any prescription. Many people use it to alleviate headaches. Morphine is a far stronger painkiller. The word morphine itself is derived from the Roman god of dreams, Morpheus, who you also may know from the Matrix movies. Now, morphine most certainly requires a prescription from a doctor. It's incredibly strong and addictive. Next up are the stimulants. Stimulants increase things in your body, like the speed of your reflexes or your heart rate. For that reason, some stimulants are used by athletes as performance-enhancing drugs. The most common form of stimulants includes caffeine, which you find in tea, cola and coffee, and also nicotine, which you find in cigarettes. So stimulants speed things up. Now, on the complete other end of the spectrum, we have the depressants, which slow everything down. By that, I mean the speed of your reflexes decreases, your heart rate drops. They don't necessarily make you emotionally depressed, but they certainly decrease how quickly your body reacts to certain stimuli. The most common example of a depressant is alcohol. The final group is quite different to the others mentioned, in that it's largely used for recreational purposes. They are the hallucinogens, examples of which include ecstasy and LSD. Hallucinogens target your nervous system and affect the way you perceive the world around you. For example, they commonly affect colour perception. So those are the 4 main types of drugs. But now let's look at their uses. Drugs can function as medicines. They can be used for recreational purposes, in other words, leisure. And some athletes use drugs as performance enhancers. In other words, drugs that enhance their performance at a particular sport. So there are 2 types of performance-enhancing drugs. Firstly, we have the anabolic steroids. These are chemicals that help you build muscle in quite a short space of time. However, they have been linked to high blood pressure, and high blood pressure is a gateway to other problems, specifically ones related to the heart. As I mentioned earlier, some performance-enhancing drugs are stimulants, those which raise your heart rate, so you can see how that might be beneficial to an athlete who wishes to pump blood around their body faster to increase their rate of respiration to get more energy for exercise. Some of these drugs, anabolic steroids and stimulants, are legal, some are illegal, but all of them are banned by sporting bodies. The 4 arguments for this are fairly obvious. For example, if you take anabolic steroids or stimulants, you have an unfair advantage in a competition. And also, some athletes may not be fully informed about the health risks of taking that drug. But on the flip side, because there's always a flip side in science, people do argue that it's the athlete's own decision. It's their body, they're adults, they can do what they want. If they want to take the risk and mistreat their body, it is really up to them. The second argument is many will say sport is unfair. Different athletes have different training facilities, different coaches, different pieces of equipment. All these give certain athletes an unfair advantage or disadvantage. So why not level the playing field by throwing steroids and stimulants into the mix? Whether this is right or wrong is entirely up to you. You decide. So that's how you describe the 4 general types of drugs and their uses. So every drug that comes onto the market will claim it can do something. Some of these claims are quite small and quite easy to believe, whereas others are quite far-fetched and much harder to swallow. Whatever the nature of the claim, as scientists we must critically look at the study that was involved behind it. So let's look at our first claim. Statins are basically drugs that claim to lower blood cholesterol. As such, pharmaceutical companies claim that statins lower the risk of heart and circulatory disease. Now, are these claims true or are these claims false? How do we decide? Well, there's only one fair way. Let's look at the study. Firstly, this study was carried out by government scientists, not by the manufacturers of the drug. This is a good thing. It means there'll be less bias, less personal investment to prove the drug works, so less likely that the results have been tampered to say what the manufacturers want it to say. Secondly, this drug has been tested on 6,000 people. That is a big sample size. This is also very important. Let's say it was only tested on one person. How could that one person represent the rest of the population? If you're testing on 6,000, however, you can bet that sample contains enough variation to represent the population fairly accurately. Thirdly, and incredibly importantly, this study had a control group. In my opinion, that's one of the first things you should look for in a study. Does it have a control group? What this means is one group were given statins and the other group were not, so you could compare the effects. Otherwise, how would you know what effect your drug was having? You must have something to compare it to. Finally, there was loads of secondary data. Many other studies did the same experiment or similar experiment and found the same thing, so other studies support the same claim. All these things suggest that we can put this into the true basket. In other words, the drug claims we believe are true. Statins lower blood cholesterol. Claim number 2, cannabis use causes mental health problems. Once again, we must look at the study behind this. While many different studies have been carried out, but the studies have shown a great variation in results. In other words, not all the results say the same thing. And also, scientists have interpreted the results differently. In other words, they've drawn different conclusions based on their results. What does this mean? Well, it certainly doesn't mean we can put the claims linked to cannabis into the true basket, but we also can't put it into the false basket, because the studies don't reach any firm conclusion. In other words, we just can't say whether these claims are true or false. So that is how you evaluate claims on specific drugs. So before a drug reaches the market, it has to jump over many hurdles we call drug trials. These are a series of many tests that ensure that the drug is A, safe, and B, effective. The best example of a drug not receiving proper trials was in the 1950s with a drug called thalidomide. It was initially intended as a sleeping pill, but then later used as a morning sickness pill for pregnant women. The idea was you take thalidomide and it alleviates morning sickness so you don't feel sick in the morning, a common symptom of pregnancy. Because they hadn't tested it properly, they didn't realise thalidomide could actually pass through the placenta into the fetal blood. The effect this had on the developing fetus was that the baby was born with abnormal limbs. In fact, in some cases, babies were born with either no legs or arms at all. In fact, 10,000 babies were affected by thalidomide and only half of them survived. The drug was obviously banned and more rigorous testing was carried out. Currently, thalidomide has been used to treat leprosy and some other diseases, including some cancers. Okay, so what hurdles do these drugs have to jump? There are 3 main stages. Trial phase 1 is the easiest, simplest, and least ethically problematic. In other words, it's unlikely to offend anyone. What we do is we test the drug out on human cells and tissues. Tissues are like sheets of similar cells. So you can see a sheet literally like a tissue paper of cells. So this is ideally where we'd like to start, but sometimes it's just not possible. For example, if we want to test the effects of drug on blood pressure or organ systems, then we can't just test it on a single cell or a single tissue because it affects a great deal more in the body. If that's the case, we need to skip right to phase 2, animal testing. In fact, British law states that before we can start testing on humans, we must actually test on 2 different species of animal. Now you can see here I've chosen to draw a rat and a frog. Rats would be a more suitable test subject than a frog, purely because frogs are amphibians and rats, like us, are mammals, so genetically we are similar. So the way the drug affects the rat's body is going to be more representative of how it might affect our body. So we test on animals firstly to find out if the drug actually works. Secondly, to find out how toxic the drug may be, if toxic at all. The more toxic a drug is, the more likely it is to damage your cells and tissues. Also, we use animals to work out a suitable dosage. The ideal dosage is using the smallest quantity of the drug possible, while still achieving the desired effects. And that's animal testing. So where do we go from here? We do the toughest hurdle to jump, the clinical trials. This is when we actually start testing on human patients. But before we do, we have to test on healthy volunteers first, because obviously people who are very healthy are more likely to be able to withstand any bad effects of taking the drug. So the best way of doing this is starting off with a very, very low dose and gradually increasing it to test the effects it has on the volunteers. Now there are 2 possible outcomes to this. Firstly, if there are harmful side effects, then we're back to square one. We need to go back to the formula of the drug and maybe change things and then retest it. But if no obvious harmful side effects are brought about by the drug, then we can move to testing on actual patients with the illness. We do this to find the optimum dose for that drug. In other words, the lowest quantity of that drug needed to still be effective and bring about very few side effects. However, it's a little bit more sophisticated than that. You know, like when you see a little child crying and they're always looking for their mum. You may notice that the mum doesn't actually treat them in any way. All she does is comfort them, and just having that intervention from someone else makes the child feel better. Well, we've kind of got to avoid that effect, very much so, to ensure that the drug is actually working. That effect of feeling like treatment's working when in fact you're not receiving any treatment is called the placebo effect, and it's incredibly important in clinical trials. So here's what we do. We divide the patients with illness up into 2 groups. So I've got 10 patients here. 5 of them are going to go into a group which will be given the actual drug, and 5 will be put into a placebo group. Now it's important to note that this test or this trial will most definitely be at least blind or preferably double blind. Blind means neither this group of patients or this group of patients knows whether they're receiving the actual drug or a fake sugar pill, which we call a placebo. A double blind trial is even better because even the doctor doesn't know which group has received which treatment. In other words, a double-blind trial completely removes bias from this clinical trial. For example, if the doctor knew that this was the group that were getting the drug and they really wanted the drug to work, they really wanted these patients to be cured, they may act differently, they may send out signals, they may give these people hope when it's just really not fair. So you've got to make sure that it is blind. So you know the only thing that's affecting these patients is the drug itself. So basically, if these patients start to get better way before these ones, you can probably assume the drug is working. If there's no significant difference between the placebo group and the drug group, it doesn't mean the drug isn't working, but it certainly doesn't mean you can sell it yet because it doesn't prove anything about the drug. And of course, if these people take longer to get better than these, it probably suggests the drug has a harmful effect on the body. Aren't you relieved now that scientists are quite fussy people? I mean, isn't that important? Personally, it gives me great peace of mind knowing that these medicines have gone through proper trials. Although it has to be said, some still slip through the net. And that is how you can explain the key stages involved in drug testing.